Welcome, everyone, to Stream B on Ecosystem Genomics here at Biogenomics 2020. My name is John Kress. I'm Curator Emeritus at the National Museum of Natural History at the Smithsonian and also co-chair of the Earth Biogenome Project. We've heard a lot about all different aspects of genomics over the last 4.5 days. And today we're going to hear some more, or primarily talks on interactions among species. Interactions among species add to the complexity of nature in a great way. And the information contained in genomes is so important in understanding the evolution and ecology of these interactions. Whether they're in gut microbiomes, whether they're in the canopy of tropical forests, or whether they're pathogens and zoonotic disease transmitters. This session will focus on examples of how ecosystem genomics can provide a more detailed understanding of these actions. We are scheduled to have 3 papers in this session. There were several other papers that were moved out of this session into others. Also, you probably have noticed that there's been quite a few talks that are about ecosystem genomics in many of the other streams and the plenaries themselves. I also want to remind you that there are 3 posters on ecosystem genomics that you should visit at some time this morning or today during the conference. At this point, we only have one of the speakers online, so we may curtail several of the speakers, but we're going to start. It's my great pleasure to introduce Adam Healy and his colleagues. Adam is from the Hudson Alpha Institute for Biotechnology in Alabama. He is going to talk to us about Sphagnum, a model organism for carbon sequestration and niche ecosystem evolution. Adam, up to you.
My name is Adam Healy, and today I'd like to talk to you about some of the genomic resources that we've been developing for Sphagnum as a model organism for carbon sequestration and niche ecosystem evolution. And before I get started, I just want to acknowledge all of the hard work of our collaborators that went into this project, particularly John Shaw of Duke University and David Weston of the Oak Ridge National Lab. There's been a lot of time and effort that has gone into this, so thank you to everyone involved. Now, just to give you a bit of an introduction about Sphagnum, you can find it growing just about everywhere on Earth. It grows in peat bogs and forests around the world. You can find it on every continent besides Antarctica. Now, despite the fact that Sphagnum only occupies about 3% of the global land area, it's responsible for storing 25% of the terrestrial carbon as peat. And typically you find Sphagnum growing in these hummock and hollow growth structures, where in a peat bog, you'll have some species that prefer to be very far away from the water table, and they grow up in these high hummock or mounds. But you have other species that prefer to be down in these low, hollow valleys that are very close to the water, or they're even submerged. Now, Sphagnum is a niche ecosystem specialist. Along this hummock and hollow gradient, you can often find 20 species all co-occurring together, growing in sympatry. Now, in addition to this, Sphagnum is also an ecosystem engineer. As it uptakes nutrients from its environment under very harsh conditions, it's able to suppress the growth of other plants and microbes that are growing there as well. Now, just to show you what this niche ecosystem colonization looks like, here you see 2 species of Sphagnum growing relatively close to the waterline. Now, as you transition further and further away from the waterline, you see that you transition to the growth of Sphagnum species A, which likes to be very, very close to the water. But as you move further away from the waterline, even a transition zone of 1 to 2 centimeters, you transition and are dominated by a completely different species of Sphagnum. As I said before, Sphagnum engineers the conditions under which that it likes to grow, but other plants and microbes cannot. And it does this through cation exchange with its environment. So as Sphagnum uptakes nutrients from its environment, it dispels hydrogen ions and acidifies its environment. Now, this creates a very anoxic conditions. That in conjunction with the secretion of antimicrobials from Sphagnum as well, it creates a very sterile environment. So sterile, in fact, that it's capable of preserving humans for thousands of years. Here's an example of the tollman that was found in Denmark peat bog that was preserved for over 2,000 years. So despite all of these tough conditions that Sphagnum generates, the peat bogs themselves are not completely devoid of life. Sphagnum actually protects beneficial microbes such as cyanobacteria from the harsh conditions that it creates. And it does this through its hyaline cells, which it uses to hold water, which maintains its internal structure. Now, the water within these hyaline cells is actually neutral pH, and it uses these encapsulations to protect the microbes that give it additional nitrogen or carbon that it can use to grow. So because of the complex interplay between sphagnum, its microbes, and its environment, the Department of Energy and USDA have been working on this SPRUCE site in Minnesota, which stands for Spruce and Peatland Responses under Changing Environments. It's a 10-year study that is used to investigate the effects of increased temperature and carbon dioxide on these peatland forests. So at this spruce site, there are these huge growth chambers that are several meters tall that they can actually use to monitor and manipulate ambient temperature and carbon dioxide and see whether or not we run the risk of these traditional carbon sinks actually turning into carbon sources under varying conditions. So to aid in this research, we've been developing some genomic resources. We have a Sphagnum angustifolium F1 population that we've sequenced to generate a genetic map. We have two high-quality reference genomes that we've generated through PacBio sequencing, and that enables really high-level comparative genomics between species. And then finally, what we want to do is take the information that we've learned from the pedigree and the references and give it sort of a phylogenetic context by looking across the major clades of sphagnum by resequencing diversity samples. So the first thing that I'll talk about now is the data that has gone into the pedigree. So generating a genetic map not only enables us to construct chromosomes within our reference genomes, but also enables us to map QTLs for carbon sequestration phenotypes that we're interested in, such as responses to temperature and pH, the changes in microbiome, and also decomposition. So the folks over at the Oak Ridge National Lab have generated a Sphagnum angustifolium controlled cross full sibling pedigree composed of about 186 individuals that we've sequenced to about 15x coverage. And we've taken all of that data, aligned it back to our angustifolium reference, and used that for SNP calling. And what we found was we got about 2.5 million really high quality SNPs at the other end. We were able to phase those that only occur within the maternal parent, giving us 1.1 million SNPs to work with. And after we filtered down those that gave us the best recombination frequency and those that were not in, high linkage disequilibrium, we cleaned the marker set down, and we got about 3,000 markers that were really great for generating a genetic map. And this was the genetic map that came out the other end from those markers that we were completely blown away with. You can see that there is, these are very, very high dense, very small gaps with 19 high quality linkage groups generated. So the total map length itself is over 5,000 centimorgans long. The average distance that you see between markers in that map is about 1.8 centimorgan, which is a great resource because when you want to use this for QTL mapping, when you want to pull candidate genes back out, the average distance among genes is about, the average number of genes within a centimorgan is about 5, with the max number being 15. So this is a really great resource for doing functional genomics down the road. But we can also take these markers and project them back onto our PacBio assemblies and use them for chromosome construction. So that's exactly what we did. So from a previous version of angustifolium that was sequenced with just short read Illumina, we saw a 100x improvement in the length of our contigs and also the number. So we got our total number of scaffolds down in the genome that was over 1,000 down to about 36. The genome came together in about 400 meg of sequence and 19 linkage groups. So we thought this was really great. So the next thing that we wanted to do is compare these genomes to each other and see what they look like. So when you compare the two references together, you can look at synthetic gene blocks between them, and immediately you see this very impressive pattern. So just looking at the diagonal in this plot. So chromosome 1 versus 1, chromosome 2 versus 2, and so forth. You can see that the two genomes are roughly 1-to-1 syntenic with each other, with two large inversions occurring on chromosome 9 and chromosome 18 between the two species. And of course, that's not what's really interesting about this plot. It's not what's going on the diagonal between chromosomes, but it's the off diagonal. It's shared synteny among separate chromosomes that look really interesting to us. And it shows very, very clear evidence of whole genome duplication events that have occurred throughout the evolutionary history of Sphagnum. And we can actually use this pattern to reconstruct the ancestral trait of the Sphagnum genome. So just looking at this pattern alone, if you just look at chromosomes 1, 2, 5, and 8, you can see that they retain synteny with one another. So you can infer that they likely have been generated from whole genome duplications from the same ancestral chromosome. So you can pair them together as such. Now, if you walk along the plot in the same way, you can do similar pairings among chromosomes and also find small little differences in synteny as well. And you can use this information to work backwards and look to see what Sphagnum looked like millions of years ago. So it looks like the ancestral trait of Sphagnum was 5 ancestral chromosomes. Now, sometime before a whole genome duplication event, a piece of ancestral chromosome 4 either broke off or was duplicated and was located on ancestral chromosome 2. The population underwent a whole genome duplication event, which gave us 10 chromosomes in total. And then sometime afterward, approximately 150 to 200 million years ago, based on previous estimates, there was another whole genome duplication event. Now, this should have given us 20 chromosomes in total, but sometime after this duplication, one of the copies of ancestral chromosome 5 was lost, giving us our modern day chromosomes that you see here. Now, because we would like to age these whole genome duplication events, and typically you do that to an outgroup. But what's interesting about Sphagnum is that it doesn't share any synteny with any non-seed plant genomes that we've investigated so far, which makes aging these events very difficult. But we can sort of peel back the curtain to see at each time there was a whole genome duplication, what sort of gene functions were preferentially retained after each event. That's what we did. So if you look at the non-synonymous mutation rate between paralogs in these ancestral chromosomes, you can see evidence of the 2 whole genome duplication events. Now, the older whole genome duplication event seemed to enrich for genes that function, whose functions are related to photosynthesis, hormone production and signal transduction, nitrogen metabolism, and phenylpropanoid biosynthesis. But more recently, the most recent whole genome duplication event seems to have retained a lot of genes that all of their functions point to protein production, processing, and proteolysis, as well as messenger RNA surveillance. And there was also genes related to circadian rhythm and also glycolysis. So it's very interesting to sort of peel back the layers to see what Sphagnum has been doing over the millions of years and what's been important to its evolution. So that brings us to the diversity samples. So again, we wanted to take all the information that was gleaned from the reference genomes and the pedigree and sort of give into phylogenetic context. So Sphagnum is divided into 5 major taxonomic clades: Rigida, Subsecunda, Cuspidata, Section Sphagnum, and Acutifolia. Now, each one of these major clades likes to grow at various pHs and various heights along the water table. So what we wanted to do was resequence about 15 species to about 40x coverage and look to see how much variation there is across all of these taxonomic clades. Each one of these genomes has been de novo assembled, and we did sort of a pseudo annotation where we took reference proteins from the 2 references, angustifolium and magellanicum. We took the protein variation and projected them into each assembly and then gathered back the variation that came back out. So we can use this information to reconstruct the phylogenetic tree and look for signatures of selection among the different clades. So as I said, we took these pseudo-annotated de novo assemblies. We generated gene trees for all the single copy orthogroups, of which there was just over 2,000 of them. And when you use that to regenerate a species tree, first thing that really struck us is that you get this nice hummock and hollow growth pattern back out, with one single exception where you see Sphagnum squarosum, which is normally part of acutifolia. It was falling out of place, which sort of broke up this overall pattern between the hummock and hollows. What we think is occurring here is that there is actually an ancient introgression hybridization that moved squarosum out of acutifolia and makes it a little bit more similar to cuspidata, which is why it's falling out of place. But regardless of this event, you can clearly see that the hummock and hollow pattern has been recapitulated in the phylogeny. So looking at signatures of selection, peat accumulation in a peat bog is typically controlled by differences in growth rate and biomass decomposition. Hummock species typically are much slower growing, but undergo slower decomposition, whereas hummock species that are down closer to the water, they grow much faster, but they decompose much more quickly. And it's these dynamics that allow peat to be deposited at roughly the same rate between the two groups. Now, when you look at what's under positive selection in hummock species, you typically find things that are related to growth and detecting gravity, flavonoid biosynthesis, and also pectic polysaccharide production. Similarly, in the hollow species, you find GO-term enrichment for genes that are under selection that are related to xylose formation and response to photosynthesis and detecting light. So it's very interesting to see that the things that modulate these dynamics tend to be related to cell wall decomposition and are likely very much affecting biomass decomposition. So in summary, what we've done for Sphagnum is generated 2 high-quality annotated reference genomes. We have created a dense genetic map and resequenced pedigree for marker trait association for interesting carbon cycling phenotypes, such as plant microbe interactions, growth rate, decomposition, and response to temperature and pH. And we've also resequenced a diversity panel along the major taxonomic groups of Sphagnum. We have assembled them and pseudo-annotated them, which has given us information on an ancient introgression that looks like it's occurred and signatures of selection among hummock and hollow clades. And with that, I'd like to thank our collaborators again, and I would be happy to take any questions. Thank you.
Adam, thank you very much for that presentation. Extremely exciting stuff, particularly way down there at the forest floor in the swamps. There's a couple questions here. I have 1 or 2 myself, so let's dive right in there. One of the questions that came over from an anonymous participant said, how deeply diverged are angustifolium and Magellanicum? The whole gene duplication must have occurred before speciation, is that right?
Yes, so we think that Magellanicum and angustifolium diverged roughly 20 million to 5 million years ago. It was actually during this time that there was a large radiation event where there was a whole bunch of diversification going on. So we're really interested to see what was going on Earth during that time that sort of caused that event.
Yeah. Is Sphagnum global, worldwide? Do they occur all across the planet?
Yes, particularly Magellanicum. You can find Magellanicum growing almost on every continent. I'm not sure if it's radiated down to Australia, but there are species of Sphagnum down there as well.
All right, another question that's come over from a participant is, can we use this, or how can we use this knowledge to improve peatland conservation and restoration?
Yeah, I think it's, well, from a conservation standpoint, most of the natural diversity of Sphagnum is located in the Arctic, which is under the greatest risk of warming due to climate change. So we really want to understand like what's going on during the interplay between Sphagnum and its microbe and its environment to sort of get a sense like what could possibly happen if things warm up too much, which is particularly what's of interest at the SPRUCE site, where they have these chambers where they heat things up and look to see how the different plant species respond.
One question, then I want to ask a question. Which method did you use to infer the introgression?
It's the quartet method. I can't think of the software specifically, but it's the 4-way test between different clades to look to see where the introgression occurred. Okay,
well, this participant might email you to get more information on that if they want. One question I had, and then we'll wrap up this session, is one of the objectives of the Earth Biogenome Project was not only to sample phylogenetic diversity across the tree of life, uh, but also to focus efforts on generating genomes in particular ecosystems. It's what we're calling the bioobservatory effort. And would generate genomes, let's say in a tropical rainforest, for all the plants, animals, fungi that are interacting to begin to understand the genomic basis of the ecology of these areas. I think I heard from Jill Wegerson's talk the other day that there is a spruce genome. So are we aiming for a bioobservatory approach in these sphagnum bogs, where we've got spruce genomes and sphagnum genomes, and what would be next genome to add there?
I think anything that's in the non-seed genome world would be great, because when we try to compare sphagnum to anything else that's been sequenced so far, we see that comparing it to itself, we see that beautiful, synteny plot where you see nice big blocks of collinear genes. As soon as we walk outside of sphagnum and try to do the same comparison, there's just nothing to see. So we really need to sequence everything to find something that's close to it, and then we can do a better job of comparative genomics.
Yeah. Okay, I think we're going to wrap up this session. Thanks very much. Well, wrap up your talk, but I also have to report that the other two speakers in this session have not appeared. I'm not sure what happened. I wasn't informed that they were not going to be available. So I think we'll just bring this session to a close on ecosystem genomics, look at the posters, and get ready for the plenary that's coming up next. So I want to, Adam, thank you for your presentation, and thank all the participants for being online. So long.
Myself, Dr. D. Mukundan from Satyabama Institute of Science and Technology, India. Before into my presentation, I sincerely thank Biodiversity Genomics 2020, welcome Sanger Institute for giving me an opportunity to share my views on my research topic, altering seawater pH and fish gut microbiome, a study based on 16S rRNA gene shotgun sequencing. This is the abstract what we have submitted to the committee. When we are talking about the climate change in ocean, we have to talk about the urbanization and the industrialization, emission from the agricultural waste, and deforestation. All these triple inputs have given a lot of carbon dioxide emission as well as greenhouse emission to the atmosphere. Because of this excessive of emission of carbon dioxide, the atmosphere become warmer, and the excessive heat and this carbon dioxide was absorbed by the ocean environment. Thus, ocean become warmer, less oxygen, and more acidic. That will lead to sea level rise, coral bleaching, occurrence of toxic algal blooms, habitat destructions, ocean acidification, and decline in the fisheries resources. When we are talking about ocean acidification, the carbon dioxide combined with the seawater, it forms carbonic acid, which breaks down into bicarbonate ions and hydrogen ion. The hydrogen ion combined with carbonate ion to form bicarbonate. This natural process has maintained hydrogen ion concentration for thousands of years, and the pH is 8.2 in a healthy, normal seawater environment. But because of this higher carbon dioxide emission, the hydrogen ion concentration is increasing and the pH is lowering. That is, it is reducing from 8.2 to lesser pH. That is not good to the ocean environment, and it is harmful to all marine organisms. Report says that it is mainly affecting shellfish animals, but it is also affecting the fin fishes, which is dwelling in the ocean environment. Thus, acidification combines with warming and deoxygenation in the ocean, and it it will leads to a lot of problem to the animals in the ecosystem. It will impact the animal in molecular level, organism level, and community level, and that will affect the ecosystem as well as the human mankind. Now we are talking about gut microbiome, and gut microbiome have an impact on the host growth, nutrition, tolerance to stress, immune system, disease, and physiology. This gut microbiome almost have a relation in all kind of organisms, including fish, wherever it is habitating. When we are talking about a fish gut or any fish in a marine environment or in a freshwater environment, the digestive tract is having the following organs or divisions in the digestive tract. It starts from mouth to gizzard, and the entire tract was occupied by the gut microbiota. We all know the digestive function is controlled by the brain with the help of endocrine system. But recent report says that gut microbiota is also playing a major role with the release of metabolites. That is also a playing role in digestive function that will control the brain. So if the gut microbiota population is affected in any means, definitely the health status of the host will affect it. Of course, this gut microbiota may affected with the changing ocean acidification or in the alteration in the pH. As I said, the internal and external factors have an impact in the gut microbiota. So the change in the seawater pH definitely have an impact on the fish gut microbiota, and that may have a greater impact on the health status of the fishes. For this, we want to study ocean acidification and their relation in the fish gut microbiome using a tool called metagenomics. We have raised the following questions. Will change in the seawater pH have an impact on fish intestinal microbiome? Will the fish intestinal microbiome play a role on environmental information processing? And will change in the seawater pH have negative impact on microbial signature? So we have done an experiment to answer all the questions. For that, we have maintained seaweed clownfish in our aquarium. We have induced ocean acidification effect in our laboratory by altering the seawater by bubbling carbon dioxide in the aquarium setup. Before entering into the experimental part, I just narrate about the aquarium maintenance. The fish maintained in our aquarium were almost the total length is 5 centimeters, and we have procured it from a fish trader. And with the proper quarantine, we have kept in the laboratory before we are taking into the experimental setup. We have fed the animals with the shrimp, boiled oyster, and clam meat 3 times a day. And the excessive materials, the remnants of the food particle, are cleaned after every feeding. The tanks were eliminated with a 40-watt fluorescence tube, and the water quality parameters like temperature, salinity, pH, and dissolved oxygen were maintained properly. The temperature is 28 degrees Celsius, salinity is 34 ppt parts per thousand, and the pH is 8.1, and the dissolved oxygen is 6.5 microgram per liter. We have maintained the light intensity of 800 lux for 14 hours light and 10 hours dark. Once a week, we have exchanged the water with 50% of 50% of new water. Thus, the fishes were maintained for 30 days in a controlled condition before the experiment. So this is the scientific classification of Sibae clownfish. The genus name is Amphibrion and the species name is Sibae. With the proper permission and egg clearance, we have maintained the fish in our aquarium setup in our lab. We have used carbon dioxide and bubbled the carbon dioxide to change the pH in the seawater aquarium. The number of fishes maintained in the aquarium is 6 per tank, and the aquarium was have a salt pH of 8.1 in the first experimental setup, and in the second experimental setup the pH is 7.8, and the third experimental setup the pH is 7.6. Salinity, temperature, and light intensity is constant. The salinity is set to 3.5 ppt, and the temperature is 23.3 degrees Celsius in all the experimental setup. Whereas the partial carbon dioxide saturation in water depends upon the change on the pH, and the days of exposure is 30 days. After the experimentation, we have sacrificed the animal, and the gut, my gut was taken as the sample, and the genomic DNA was isolated with the help of DNA mini preparation kit, which is available commercially. We have done 16S rRNA gene amplification with PCR, and we have constructed the gene library, quantified it, and we further went for sequencing. With the proper bioinformatic analysis, we have done chimera detection, statistical analysis, and comparative analysis. For this purpose, we have used Mathur software, which is available online. When talking about the results, this rare fraction graph shows that the goodness of our sequence, what we have analyzed, and it have 97% similarity cutoff value. And this is the overall data what we have obtained from our sequence. In the experimental tank of pH 8.1, we have almost analyzed 38,166 sequence with a goodness coverage of 97.32%. In the experimental setup of 7.8 pH experimental tank, we have analyzed 31,303 sequence with the goodness coverage of 97.15%. And the experimental setup with the seawater pH of 7.4, the sequence analyzed were 37,412 with the sequence coverage of 97.06%. And we have also analyzed the controlled seawater environment. In seawater, the number of sequence analyzed were 36,456, and the goodness coverage is 97.27%. Almost we have got more than 1,000 OTU in all the sequence, and the OTU range were given here. We have also done community richness analysis and community diversity analysis with the Chomsky Simpson Shannon experiments. And these graphs shows the alpha diversity and beta diversity, and this genetic distance of 3%. This color code represents the bacterial population variation at the class level, family level, genus level, order level, and phylum level. Almost we have given the color code for all level of the taxonomic position for the experimental setup with the pH of 8.1, 7.8, and 7.4, and we have also given the control results also here for comparison. As I already said, we have used Mathur as well as ribosomal database project from internet to do the entire bioinformatics analysis. This dendrogram shows the difference for all the experimental setup as well as for control at phylum level as well as at genome level. This shows the variation in the bacterial population of the gut microbiota. This Venn diagram shows the uniqueness of the OTO for all the experimental groups as well as for the control, and almost more than 13,000 unique OTUs were identified in each groups with some variations, and a common OTU of 200 were observed. The OTU at species level gives some important data when we have got the results. The gut microbiota is almost occupied with bacillus species. That is good. That will help. That will be a probiotic in digestive tract, and it is very good to the fish internal gut environment. But we have also observed some Vibrio population in the gut. And when the pH is lower to 7.4 in the experimental tank, this is the observation we saw in the Vibrio population. There is an increase of O2 in Vibrio alginolyticus, Vibrio harvey, Vibrio medrasius, Vibrio parahemolyticus, and Vibrio ponticus, and Vibrio species. As we all know, this Vibrio population will have a negative impact on any kind of aquatic organism, particularly here in the case of seaweed. Definitely, this Vibrio population will lead to vibriosis, disease that will lead to any kind of aquatic organism. This shows that lowering pH, that means the effect because of this ocean acidification and the alteration in the pH definitely have a negative impact in the microbial signature of the gut microbiota, and it have to be studied further in this point. And we have also done some functional analysis, use the metagenome what we have obtained with the help of the PyCrust software using Galaxy platform, and we saw that microbial metabolic pathway, particularly that is reflecting on environmental information processing, especially in membrane transportation metabolic way. This is because of the excess of carbon dioxide, excess of hydrogen ion in the seawater, as well as the bicarbonate ion, which is playing a major role in the ozone acidification. Our questions were answered with our experiment. Yeah, the intestinal microbiome have an alteration in its signature, and all these ozone acidification effect have an effect, and it reflects on the microbial metabolic pathway system, particularly in membrane transportation process, and they have a negative impact by increasing the Vibrio population in the gut environment. Already, some of the studies have already reported by Stump et al., Sylvian et al., Hu et al., Ambulkria et al., Adamski, and Khan et al. On 2013, 16, 17, 18, and 2020. They say fish gut pH regulation is sensitive to ozone acidification. Here we want to report that this change in the pH in the fish gut and the environmental change, particularly like ozone acidification, have a great impact on the fish gut microbiota. We all know that the gut microbiota population, if it is altered, that will have a negative impact on the health of the host, and that will be reflected here in our experiment with the increased population in the vibriosis. Maybe this kind of experiment have to be studied more, as the relationship with the pH and microbiome relationship have to be studied in detail. And almost in our study also we need some alarming we found out that it is an alarming indicator and it have to be studied further in detail. The take-home message is, we have to control the carbon dioxide emission in our atmosphere, because it have a greater impact now, even in the microclimatic change, like even the gut microbiota population is getting affected. So we have to stop emission and we have to love our nature. Thank you.
So, hello everyone. This is Ashok signing in myself from Tamil Nadu Agricultural University, Coimbatore, India. So, I'm here to present in this wonderful Biodiversity Genomics 2020 with the topic of genetic characterization of maize hosting Spodoptera frugiperda, the most serious invasive alien threat to the maize crop in India, by using the PCR RFLP techniques. Let us see the brief introduction about this particular pest. So, since we know from the day of introduction, there are two strains which was reported in the entire world. One is R strain that is feeding on the rice, and the C strain that is feeding on the corn, that is the maize. Here we have just mapped the potential, the native, as well as the recent, the particular host of entire country where it belongs to. So we know it was first reported in 1950s by Luginbel in America. After that, Gauguin in 2016 in Africa, followed by 2018 in Karnataka by Sharna Bashappa and co-workers. So, which race is actually in the race and why does it matter in India? Of course, the real thing which we are going to see. So, the methodology which I have followed was, it was a simple DNA extraction protocol by using the CTAP technique. Here, our experimental detail covers three sets of primers, one with an universal primer and another with an the same COI targeting region, and another one is the TPA region where the three set of primers were involved. And it is a simple DNA extraction protocol by using the CTAB method, followed by the restriction fragment length polymorphism technique by using two set of restriction enzymes. One is the SAC digestion and another one is the MSP digestion, which are the strain specific. So let us see the results. The first one is the genetic characterization of using the COI primers and the sequence identity matrix as well as the sequence difference code matrix. So here there are two genes which we have involved targeting the COI region. One is an universal primer and another was another set of COI primer, whereas both gets amplified with their base pair region gets differs. Among that, the sequence identity as well as the sequence different code matrix denotes our particular thing what we are having matches exactly with the African population, that is the sequence identity matrix, and we have 100% matching with the haplotype R which was reported in the GenBank sequence. Followed by the sequence different code matrix denotes our rice strain does not have any deviation in the nucleotide, but our strains which have differ with the corn strain with the set of three different set of nucleotides. Next one is the restriction mapping. Here, the restriction mapping denotes the SAC digestion is present, which denotes the SAC digestion is specific for the rice strain. So, the SAC strain, which involves GAG-CTC nucleotide, which is present at 370 base pair as well as on 154 base pair, which was denoted in this GIST map. And here, the bottom of the PCR RFLP gel image shows the restriction fragments. Next one is the genetic characterization using TPA primers and the restriction map of that particular TPA MSP region of CCGG. Of course, our particular gene contains MSP, which denotes the MSP is specific for both R strain as well as T strain. But if you want to concentrate on the C strain, you have to target the T10 region. So this proves an additional confirmation that our particular sequence, what we are having was an rice strain. Next one is, here is a nutshell which showing the TPA and the two COI region, which denotes the restriction fragments, whereas the TPA denotes the restriction is present in the case of SAC. It is not present in the case of SAC, but present in the case of MSP, whereas the COI region denotes the restriction is present in the case of SAC and not in the case of MSP. This proves that our strain was below our insect which hosting the maize, which belongs to the rice strain. And here we have mapped with we have done a phylogenetic tree which targeting the COI gene as well as the TPA gene, whereas our Coimbatore population which matches similar and forms the same clade with the particular COI gene in that rice strain. as well as here is another restriction map which shows our Coimbatore population, which matches with the rice haplotype. So finally, I would like to say that PCR RFLP technique will help to solve the problem of cryptic strain, as well as the COI and TPA gene segments determine the isotype with specific to that of haplotype. And with this denotes the population migrated from USA to Africa, and Africa it migrated to our India. The strain variation alone will help to properly address the problem of monitoring as well as the integrated pest management. The future direction involves, since we are in the stage of population genetics, we have done the chemical ecology and the climate change impacts of this particular strain specific fall armyworm, and we would like to give up a model which would like to help, because that particular strain will differ with the pheromonal compound. So we will design a pheromone product that will help the farming community for sure in future. Finally, though there are several advantages, the gray area involves it denotes only the Coimbatore population and need more survey location to get further confirmation. So finally, I would like to acknowledge the Department of Science and Technology for their financial grant in this project and my advisory team for their wonderful support throughout this research. Thank you, and thanks to one and all for your kind cooperation and listening.